Hi everyone, and welcome to this introduction to WuXi AppTech. My name is Dave Madge, and I'm part of the WuXi team in Europe, based in Cambridge in the UK. To orientate those of you who may not know our company, we provide a broad range of research, development, and manufacturing services that enable diverse discovery organizations, large and small, across all therapeutic areas and modalities. We're a trusted partner to over 4,000 discovery teams working to identify new therapies globally. Our company was founded just over 20 years ago at a single site in Shanghai, and today extends to more than 30 sites, supporting over 27,000 employees. And I'll show you a little about how our teams are distributed on the next slide. China remains our center of gravity, with more than 15 sites, mostly to the east of mainland China, supporting a wide range of discovery, chemistry, and biology, as well as small molecule and large molecule manufacturing. We recently opened a substantial new site in Chengdu, towards the west of China, capital of the Sichuan province, and famously home of the giant panda. In the US, we have sites for discovery, chemistry, and biology, medical device testing, toxicology, DMPK, and clinical development services. The US is also home to our cell and gene therapy manufacturing and testing division, an activity recently expanded in the UK with an R&D site in Oxford. Elsewhere in Europe, we also have a site in Germany for biophysics, protein engineering, and crystallography, and a small molecule manufacturing site in Switzerland. Our philosophy and strategy is to build platforms and collaborations that enable as many organizations as possible to pursue their ideas towards solving the very well-known challenges that the healthcare industry faces. We work to shorten R&D timelines, to make R&D more cost-effective, so that affordable treatments can be found for more and more diverse diseases. In order to achieve this, our solution is to build and provide to our collaborators end-to-end discovery and manufacturing capabilities. In particular, we're supporting small molecule discovery and manufacturing, drug and medical device safety testing, clinical research, and cell and gene therapy research, testing, and manufacturing. Our chemistry and biology discovery platform encompasses all stages, from target validation with gene editing tools, through creation of bespoke assays and hit finding with multiple platforms, through to lead optimization supported by in vitro biology, in vivo biology, and DMPK. This platform extends further into late stage and IND-enabling toxicology studies and GMP manufacturing formulation. So we are routinely using gene editing and protein engineering to validate targets and produce cell lines and proteins for assays and structural biology, working across all target classes and therapeutic areas. To date, we've produced well over 1,000 assay or crystallography grade proteins. I'm going to focus down on our platform for immuno-oncology drug discovery over the next few slides, an area in which we have over 400 scientists. This team supports in vitro and in vivo efficacy models, sequencing activities, including single-cell sequencing, biomarker discovery, and support to clinical studies. So to look at a typical workflow in this space, we might start with target identification or validation with gene editing, move on to in vitro immunology assays, and perhaps in vivo pharmacology with Syngeneic or Humanized models. Along the way, we might use FACS, immunohistochemistry, and sequencing to examine in detail the functional effect of molecules at a cellular level. And in doing so, potentially identify biomarkers that we can translate into the clinical setting. So to look at each of these areas in a little more detail, we have a team of nearly 50 scientists working with gene editing tools for target identification and validation, both in vitro and in vivo, including working with cancer and immune cells and carrying out CRISPR screening studies. Our immunology platform is comprehensive, encompassing cell activation and differentiation assays and immunosuppression assays. I illustrate here some data from T-cell exhaustion assays, mixed lymphocyte reaction assays, assays in myeloid-derived suppressor cells, and antibody-dependent cellular toxicity assays. We also have a useful panel of Syngeneic and Humanized models with extensive standard of care data, as well as reference data with many classes of oncology drug, exemplified here with checkpoint inhibitors alone and in combination studies. Adoptive transfer tumor models using human PBMC provide reliable translational data, and we've validated models for targets in hematology, gynecology, urology, GI, and lung cancers. We can also knock in human targets into genetic mouse models, shown here for a collection of major IO targets, including into C57 and BOLB-C mice. And this can be extended to accommodate double and triple knock-in studies. Our technology platforms for cellular profiling, helping to understand mechanism of action and to develop biomarkers, encompasses fluorescence-assisted cell sorting, immunohistochemistry, migration inhibitory factor assays, single-cell RNA-seq, and nanostring platforms. These platforms, of course, form the basis of our clinical biomarker support, providing CAP-certified and GCP-compliant tumor tissue profiling by pathology and FACS analysis, for example, and employing the HALO digital pathology system. To date, we've supported well over 100 clinical biomarker studies. Turning now to specific platforms for oncolytic virus discovery and testing, we're working in particular with AAV, such as H1N1, and HSV virus strains. We support production and engineering of the virus, in vitro efficacy in tumor cells of many types, and in vivo efficacy, distribution, shedding, and combination therapy studies. To provide some examples of ongoing projects, we're working in MC38 adenocarcinoma cells, U87 and GL261 glioma cells, and various hepatocellular carcinoma models with a mixture of HSV and AAV delivery. I show here examples of tumor growth inhibition with anti-PD-L1 delivered by HSV in an MC38 model on the left-hand panel, and a test article inserted into AAV in a HEPA-1/6 model in the right-hand panel. On this slide, I show some adenovirus H1N1 models currently under development, encompassing models for pancreatic, liver, colon, and breast cancer, and sharing some preliminary data from pancreatic cancer and liver cancer models using H1N1. I mentioned a little earlier our broad client base, well over 4,000 clients around the world, many of whom are working with new modalities such as bifunctional molecules, antisense oligonucleotides, cancer vaccines, macrocyclic peptides, and cell and gene therapy, and many more. You can see that our clients range from academic teams, non-for-profits, small biotech, through to global pharma companies from all corners of the world. It just remains for me to thank the organizers for the opportunity to discuss our platforms and to thank you for your attention. Many thanks.